And Cameron is going to be talking about oxycodone addiction model behaviors following constitutive reciprocal Zhx2 gene editing versus adult brain liver viral overexpression BALB-C substrains. Okay, thanks, Cameron. Take it away.
All right, thank you to the organizers for allowing me to talk today. I am now a Husky. I used to be a Boston University Terrier, so Terrier, right? Yeah. So it's definitely an improved mascot. So it's short talk, so I'm going to thank everyone first. Primarily Will and Sophia, who generated most of the data I'm going to show you. Jake Byerly, who's in Vivek Kumar's lab now, he led us to this candidate gene, Zhx2. Marty Ferris, who did a whole genome sequencing short read on the BALB-C substrains, various funding sources. Actually, the project funded from Gary Peltz led to the identification of these BALB-C substrain differences in oxycodone phenotypes. So with that, let's get started. So oxycodone is a big part of the opioid crisis. Notably, in 2010, a lot of people don't know this, the abuse deterrent formulation was approved, and you can see it's much more difficult to crush up, so you can imagine more difficult to inject, more difficult to snort. This led to the surge in heroin. This no longer had any street value. And then, of course, with heroin came fentanyl, which is kind of this portion of the curve here. So oxycodone remains highly misused. Prescription levels have declined, but it is still being misused. It's metabolized, interestingly, into this much more potent and efficacious metabolite called oxymorphone. It's a minor metabolic pathway, but on the other hand, it's up to 40 times higher affinity. So this is potentially a major component to opioid therapeutic effects and potentially addiction liability. This is a familiar story. So with codeine, the active metabolite is actually morphine. And so this is mediated by the CYP2D6 pathway, cytochrome P450s, which is also involved in the production of oxymorphone. So with this information in mind, I'm going to introduce you now to some of the interesting findings we have at the behavioral level and with the oxymorphone metabolite level. So we started working with these closely related substrains, BALB-Cs, a few years ago. The main advantage, which I think has been pointed out in at least one other talk, is that they have orders of magnitude reduced genetic complexity. And this can frequently make identifying the causal genes doing QTL analysis much more efficient and straightforward. So I'm going to show you a lot of data focusing on this timeline here. I've highlighted the oxycodone treatment days. And so when you look at locomotor activity, you'll see a seesaw. The lower days are the saline, higher days are the oxycodone. The saline controls will be flat across the board. So what we found was, if you look at state-dependent condition place preference, so that is if we paired oxy a couple of times, and then under the influence of the drug, we asked the mice which side they like better. What we see is a robust strain difference, specifically in the females. And you'll see that the BYJs don't show any state-dependent reward expression, but if you give the J females, they make a beeline for the oxy-paired side and show a very strong preference. And so this got me thinking, perhaps there's something to do with entry into the brain, pharmacokinetics, that could be serving as a cue to mediate this behavior. We didn't see it in the males, notably. And so I asked Jake to start collecting brains so we could look at oxymorphone levels. And lo and behold, we saw this interesting pattern that kind of matches up with the state-dependent reward expression. And that is the females had the highest level of this highly potent active metabolite oxymorphone. And so, at the time, we didn't take any livers, so our expression QTL data was limited to brain tissue, striatum, and hippocampus. But what we found to corroborate this QTL for oxymorphone levels in the brain was an expression QTL within this locus, and there was only a single eQTL that mapped to the same marker that peaks with the whole brain oxymorphone concentration. And so this is a transcriptional repressor. I'll tell you a little bit about in a minute. And you can see the effect plot according to gene dosage or allele dosage nicely recapitulates and matches the parental strain difference. And it's driven by the females. So we're confident that this locus contributes to the majority of variation in oxymorphone levels in the females. So Zhx2 Not a lot is known about it in terms of its function in the brain. It's been highly studied in the liver. It's implicated in liver and kidney cancer, involved in cellular proliferation and differentiation, immune neuronal liver development. Notably, though, it's involved in metabolism of lipids, and it shows sexually dimorphic expression of cytochrome P450 enzymes in the liver. So the idea is that normally it represses the expression of these cytochrome P450 enzymes. We didn't identify this, but several years ago, Jake Lewis's lab and others, I think, identified this mouse endogenous retroviral insertion between exon 1 and exon 2. It's about 5.6 kb long. And this is present in the J's, and so it leads to reduced expression at the mRNA and protein level. And you can see here that in the J strain, none of the short reads line up to this sequence here, whereas all of the BYJ ones do. So we've nicely replicated that in the short read data. And since then, long read data has come out from the Zhx lab, again, identifying this mouse endogenous retroviral insertion. And so, so this is our candidate. And hold on a second, did I go back? No. Yes, this is our candidate gene. So what we did next was we took a variety of approaches to target this gene. First, we wanted to remove the MERV and restore normal Zhx2 expression and function. And the second thing we did was knock it out. So we excised exon 3 using CRISPR. This is the only coding gene within Zhx2. And then finally, we're using AAVs to overexpress on the J background, the ones that have the MERV, the reduced expression, to rescue or restore Zhx2 levels in adult mice. So we've been targeting both tissues. And you can see at the RNA and protein level, we've nicely recovered gene expression with the knock-in. This is getting rid of the MERV. And then with the knockout, you can see that we've completely eliminated at the RNA level. And then this is nicely recapitulated at the protein level. And notably, you'll see the CJs here. It's not a full, the MERV doesn't lead to a full knockout. It leads to about a 50% reduction in expression. And so the overall hypothesis, this framework here, is that the MERV is leading to reduced expression of Zhx2. This is turning the brakes off, getting rid of the repression of cytochrome P450 enzymes. Increased CYP2Ds will lead to increased brain oxymorphone levels and increased addiction model behaviors. And so to keep this in mind, I'll keep showing the same guidepost to come back to. So again, this is locomotor activity across days. The higher days indicate the oxy days, and you can see, much to our dismay, with the MERV excision, we did not see any robust statistically significant phenotypes. Power analysis told us we needed approximately N of 14, 80% power. And so some of these trends, like we've just kind of, left it at that. So I wanted to present the bad news first before I move on to the good news. And so with the knockouts, actually, we kind of got what we expected. So remember we moved exon 3, and what we were predicting was mimicking this MERV, this loss of function, should then turn the brakes off cytochrome P450 expression, increase OMER levels, and increase behavior. And so that's what we see in terms of oxy-induced locomotor activity specifically. So you don't see this on saline days, but you do see it in response to oxy. And this is in the females. In the males, although we don't see any significant differences during locomotor training, what we do see is an increase in preference, state-dependent learning that I talked about earlier, this sort of drug cue-induced reward expression. And so just to further hone in on these significant results, I just want to show them because they're very beautiful. This is the female data, the locomotor in response to acute injection of oxycodone. And nicely, CPP has a lot of naughtiness to it sometimes, but I just wanted to be upfront here and show it to you in multiple ways. If you look at the effective day and interaction with genotype, you see a nice increase in the males. Across time, you can see beautifully there's an increase in time spent on the drug card side. If you do the subtraction measure, you also get a significant interaction. It shows increased preference in the knockout males. And then finally, across time, this is looking again at the subtraction data. So we're very confident in this result, and Will is very happy to have gotten something from testing this many mice. So the next thing we did was liver overexpression. And I won't go into the details of the construct. Suffice it to say, it is specific. We don't see overexpression in the brain. We inject it intraorbitally. Let's say it works pretty well, 100-fold increase in expression, not bad. And so the prediction was that increasing Zhx2 expression should restore this repression activity, lead to a reduction in brain oxymorphone levels, and a reduction in addiction model behaviors. And so we have some evidence for that, thankfully. In the females on day 2, we see a decrease in acute locomotor response to oxy. In the males, we see it on day 4. Perhaps unexpectedly, though, what we see in terms of expression of state-dependent learning is an increase in the females. We'll take it. I mean, it's still a phenotype, so it's pretty robust. And so just to hone in on these significant results again, they're quite nice. You can see that our predictions were correct. We see a reduction in oxy-induced locomotor activity with this liver overexpression. And then for the females, we see this nice enhancement of state-dependent reward learning. Okay, unfortunately, though, however, we did not see any difference in any of the metabolite or the drug levels within the brain in the females. This is disappointing. I have some potential explanations for why this might be true. Get to it later. However, though, in the males, we did see exactly what we predicted, was a reduction, particularly in oxymorphone levels with this liver overexpression. So supporting the hypothesis that this is leading to repression of cytochrome P450s and ultimately a decrease in OMOR production. And just to remind you, this is all in the brain. And we're just starting to kind of characterize the expression that we got within the brain. So we did these lateral ventricle injections at 10 microliters. We waited 3.5 weeks before we did the phenotyping. And this is just to show you a few sections from striatum and septal nuclei in the hippocampus. What we saw, and I think this might be due to the BALB-C strain, is the expression tended to be localized near the ventricles. So it didn't really penetrate the tissue too far. This is not what you typically see with a black 6 mouse when you do these AAV overexpression studies through the ventricles. But nevertheless, we did get some phenotypes. And so I think this is kind of a blessing in disguise if we choose to go forward with identifying the brain regions and cell types. And so what we've seen so far, and we've done this once, 16 controls and 16 of the overexpressions, we see, as we predicted, did we predict this? Yes, a decrease in oxy-induced locomotor activity, and this is correlated with place preference. So far, only the oxy-induced locomotor activity on day 9 during expression of preference is significant, but you can see things are trending in the same direction. And indeed, with the acute injection during training, we also see a reduction. So, so this does support our hypothesis that we originally generated. And then again, looking at OMOR levels in the brain, we do not see any statistically significant differences or even trends in these metabolite levels, so failing to support our hypothesis. So what can we make of this? So what I've done here is I've shown in the green what we predicted that worked, the red what didn't work, and the brown which kind of worked but went in a different direction. And so luckily it's not all red. And so you know, I think the main point is that the predictions that were with regard to acute drug-induced effects were largely accurate. So these acute oxy-induced locomotor activity phenotypes, I think the effects on these more complex traits like conditioned place preference and ultimately intravenous self-administration are going to be more complex. Very difficult to predict these complex behaviors off of metabolite levels in the brain. So, yeah, probably running out of time. So I just want to get into some discussion points. Validation is hard. So I'll note that these substrains are nearly genetically identical. There's only about 8,000 variants that segregate between the two. So you can imagine if you're using something like diversity outbred, it could be a nightmare to make predictions. And what background do you use? So, you know, sometimes the reduced complexity has very straightforward predictions that turn out perfectly. Sometimes they don't. Sex-specific effects, I think, support the effects of this gene and perhaps its causality with the QTL. even though the QTL was actually for brain oxymorphone levels. These various manipulations, looking at overexpression in the brain, may not be enough. It may require both the liver and the brain to get the phenotype that we're hoping for. And so we're still very much pursuing this hypothesis. And then finally, very little is known about Zhx2 in the brain, so we want to identify the transcriptional targets in various cell types and move on to more complicated behaviors like IVSA and ICSS during withdrawal. And then I've listed alcohol here, not for me. We did recent proteomic analysis and identified differences in alcohol enzyme genes. So we think that there's a varying domain alcohol drinking phenotype here too. And I knew this was going to be too long, and that's okay. So yeah, probably no time for questions, but thank you.